Hey, we'll get started. Welcome back for the second day. Our first speaker is Dr. Ben Maoz from Aviv University. He studies brain physiology and brain injury through tissue engineering. His title is Organ on a Chip, a new tool for studying human physiology. Dr. Maoz?
Hello, good morning. Here we'll share my screen.
Yes, we can hear you.
Great, so all of you can see me and all of you can hear me. So first of all, I would like to thank the organizers who actually invited me to this really amazing event. I heard all the talks yesterday and it was truly amazing and inspiring. I would like personally to thank all of you who are early birds and woke up so early to hear the talk today. I really wanted to come in person, but unfortunately I had some personal matters, but I promise I will come soon. So today I'm going to talk about organs on a chip. And so far, the conference was highly focused on IPS. And I would like to show you that what I'm going to talk about today is basically like a yin and yang, is really complementary to all the talks that you saw until now. Because as you saw, IPS involved human tissue to model disease. You can do genetic modifications and to identify genetic alternations and many, many, many more. On the other hand, organs on a chip actually enables to study organ-organ interaction, to induce physiochemical cues, to mimic the in vivo microenvironment, and again, many additional things. So the part of my talk in this bigger picture of the symposium is basically to show you new tools that allows you to take the IPS models one step forward. So let's take a step back to the future, and let's see basically why organs on a chip started, or actually why do we need organs on a chip. So as you all know by now, drug development is a long and tedious process. It takes up to between 10 to 20 years, between $1 to $2 billion to develop a drug, FDA-approved drug. And all of us are familiar with this process, which starts with thousands of compounds. We test this on tissues. Then we go to preclinical trials with rodents. Then we go to clinical trials, and we finish with one FDA-approved drug. But what we need to keep in mind is that between 60 to 90% of the drugs that actually successfully pass animal trials fail in human clinical trials. So just to stress this number, imagine that you have a rodent who has cancer and we developed a drug. It took us between up to 6 or even 10 years, or it costs something like $500 million or even more. And this drug works perfectly on the rodent, but once we give the same drug to a human, between 60 to 90% chances that this drug will fail. And why? Because most humans are not rodents. Or in other words, there are many differences between rodents and humans. Therefore, the ideal model will be to work with human model, but obviously we cannot just take a human being and to try our drugs on it. So what we are trying to do in the lab is to create a human model that is not a human being. The way we do it is basically a new concept known as organs on a chip. And the idea is basically to take a human tissue and to mimic this, and to mimic the functionality of a specific organ, and to put this inside a microfluidic chip. For example, the kidney is a filter. And if we take human kidney cells and we create something that works like a filter, and we put this inside a microfluidic chip, we will call this a kidney on a chip. Now, let me give you an additional example. The heart is a pump. So imagine I take human heart cells and I create a pump out of this, put this inside the microfluidic chip. I will call it a heart on a chip. And here you can see human heart cells that we put on a gel. Every time we give electrical stimulation, you can see that the heart cells contract, and therefore they contract the gel. We can monitor the amount of force and shear and stress that's being applied. We can induce different electrical stimulations, and then we can add different drugs and to see how the cardiac tissue is actually responding to the drugs or the disease we induce on the tissue. So here are two examples. One is the kidney on a chip and the heart on a chip, but we can do it for many more organs, as I will show you later. And before we will move on, let's go again a step backwards to where it all started. So the first organ that was developed by Dan Hu and Don Eberg at the Wyss Institute at Harvard University. This is where I did my postdoctoral studies. You can see the chip actually has two channels, which are separated by a membrane. On one side, you have lung cells. The other side, you have the capillaries, the endothelium. The chip itself is elastic, so you can induce forces which mimic the breath movement. You can have air on one side. You can have blood at the other side, which is relevant to one of the talks you will see later on today when they will talk about the gut. And we can do more sophisticated things, such as to have bacteria on one side of the tissue. We can have white blood cells at the other side of the chip, and we can actually identify how the white blood cells penetrate through the tissue and actually tackle the bacteria. So what we actually did in the lab, we were able to create and link up to 10 different organs. The organs actually, we can link them to each other. As you can see, we have the brain, the BBB, the lung, the heart, the muscles, the gut, the liver, the kidney, the bone marrow, and the skin. And the first question usually that I've been asked is, well, it's a great concept, but actually, how good is it? How clinically relevant this system is? So here you see in the dotted lines, here these small dots, you can see that, here I'll show you with the laser here. So here you can see with the dotted lines, these are basically, this is data that we achieved from our linked system about the drug concentration of cisplatin that was perfused in our 8-organ system. And the squares that you see here is data that was taken from the clinic. And as you can see, the data that we got from our system is very, very similar with high correlation to the data that was achieved in the clinic. So actually, we can say that there is a very high correlation from what we can get in our system and what we see in the clinic. What we actually see here, we look at different parameters relevant to drug, to drug characterization, such as the PK/PD and the different levels of drug. We identified the blood after it was metabolized in the liver, and we had renal clearance from the kidney. We can see here another example. So we, in addition to cisplatin, we worked also with nicotine. Again, the dotted lines is the data that we were able to get from our system to the linked organ linked system. And you can see here the squares is basically the clinical data. And again, you can see high correlation, and you can see that actually there is a clinical relevance from what we can get from the linked organs on a chip and the human clinical data. So the next question usually that I've been asked is, okay, what are the readouts? What can I actually get from such organs on a chip? So I will start by saying that all the regular biochemical characterization that you do in your regular setup, you can do as well in the chip. We were able to do metabolomics, proteomics, sequencing, and other things. But when we talk about the 8-organ linkage, you can see here that each organ has a specific tissue, a specific functionality, and therefore a specific readout. So in the 8-organ linkage experiment, for example, we were able to do immunohistochemistry of the endothelium, gut endothelium. We were able to do immunohistochemistry of the gut, and we were able to measure permeability of the gut. Here is permeability values. This is 1 week that they were linked, the second week they were linked, and the third week they were linked. We can link the organs up to almost 2 months, but in this experiment, we did it for 3 weeks. For the liver, again, we can do immunohistochemistry of the liver. We can look at the albumin production during the 3 weeks. Kidney, we can look at the kidney, the renal clearance, at the albumin reabsorption. Lung, we can look again immunohistochemistry, and we can look at the permeability of the lung. Heart, again, immunohistochemistry, and we can look at the LDH, and I will show you later. We can also look at the functionality of the heart. We can look at the skin permeability, the blood-brain barrier, LDH and permeability, and the brain. We can look also at the glutamine-glutamate ratio and the electrophysiology. So how we can actually do it, we can take regular organs on a chip, we can make them as a smart organs on a chip, and by that I mean that we can add different sensors. So we can have these two sensors here that we can measure the barrier function. It is dependent on the impedance. So if the impedance is changing, we can actually monitor the barrier functionality of the endothelium. We can measure electrophysiology. In this case, the electrophysiology of cardiomyocytes, but we can do electrophysiology of neurons as well. And here is an example. So you can see here that we have a barrier here where you have the control. So you can see here a barrier. Here you have holes in the barrier. And when the days goes on, you can see that as the day goes on, you can see that you have less holes and the barrier is getting better and the impedance is increasing. Now you don't have any holes and you see you get to this plateau. We can also do electrophysiology. So here we measure actually the contractility of the cardiomyocytes. And we can induce drugs, for example, TNF-alpha. The TNF-alpha is inducing holes in the barrier. And here you can see we can measure this in situ. This is the control. It continued to be high, but you see that here suddenly you have a very significant decrease on the barrier functionality after we add TNF-alpha. And we can add also isoproterenol, and we can measure the electrophysiology of the cardiomyocytes, and we can see how the contractility is significantly being increased. So we can do many, many characterizations by using smart chips as well. So at this point, I show you that we can link multiple organs, that we have multiple sensors in the chip, but I would actually like to show you some examples of the things we do with specific organs. And the first thing I would like to start is an example that is actually close to my heart, which is a heart on a chip. So what we can do with a heart on a chip, this is usually how we look at cardiomyocytes. We have cardiomyocytes in a dish. It is a 2D system, but the real heart is a 3D system, and all the time it contracts against external force. What is the external force? Is the blood pressure it needs to contract against. And the question is, how can we actually mimic this in vitro? So what we did is actually to culture cardiomyocytes on top of this gel that you see here. Every time we give electrical stimulation, this gel is contracting because the cardiomyocytes here contract as well. This is great. This is nice, but it doesn't mimic the forces that are being applied on the tissue during contraction. In order to mimic this in vitro, what we did is the gel that you see here is brown due to the fact that we embedded many magnetic nanoparticles inside the gel. So every time we come with a magnet, and this is what you see here on the left, we pull down the magnetic gel. So here you see the magnet goes up and the magnetic gel go down. So every time we want, we can apply force by pulling the magnet down. And when we actually combine the two, we can induce electrical activity, which creates contraction on the cardiomyocytes, but we can induce force by pulling it down with a magnet, and we can induce force which is opposite to the direction of contractility. And how relevant it is to the clinic? Well, again, here, usually when you want to test a drug in the clinic, you are looking at values that are related to PV loops, the pressure volume loops. And for this, you need to induce preload and afterload. And this is something that is and by using our system, we can actually induce preload, we can induce afterload, and we create these PV loops in vitro. As you can see here, we have PV loops as a baseline. We can add the drug such as digoxin, and you can see immediately the difference between the the baseline and the preload and afterload when you add this drug. So again, we can actually build platforms that we can correlate the in vitro data we have to the clinic. So this is the goal of our lab. Now I would like to give you another example from all the 10 organs that we work with. I would like to give you an example with the organ that we rarely use. And obviously I'm talking about the brain. Okay, so the example here is that here what you see in this amazing video by this video was taken by Ed Boyden with the method he developed named Clarity at MIT. And what you can see here is basically an amazing video of many neurons that you can see in half of a rodent brain. What I want you to notice is, except the complexity and the amazing number of neurons that are organized in a very impressive way, I must say, I would like you to keep in mind that what you see here is actually half of a rodent brain. Or in other words, you actually see half of this. What I want you to keep in mind is that for most humans, this is the actual brain and the actual size. And the human brain is unique not only with its structure, but also with its functionality and disease, which means that if we want to study disease, which is human relevant, we need to work with human relevant systems. And therefore, we try to create a brain on a chip, which mimic the neurovascular unit. This is the neurovascular unit. It is actually the system that links the brain vasculature and the neurons and the brain vasculature and the parenchymal. And the way we created the neurovascular unit on a chip was to take this complex anatomical system and to break it down using the chips. So we created a blood vessel chip, the BVB chip, the brain chip, which mimics here the neurons, and the vascular chip, which again, it's the BVB, and like this, we can actually mimic the lymphatic system. What is really nice is that everything that is being secreted from the vasculature is going through the parenchymal, is interacting with the astrocytes and pericytes, interacting with the neurons, and then it goes out, and you have cell-cell interaction using this system. And here you can see the actual image of the cells. You can see 3D cells of human brain microvasculature in a lumen structure. Here in blue and yellow, you can see the pericytes and astrocytes, and here you can see the human brain neurons, and similar here. So what we can do with the system, we can actually test how different toxicants affect the brain, how disease is affecting the brain, and how different drugs affect the brain. So if I already mentioned drugs, and I mentioned the fact that we can take this complex anatomical system and use the chips to break it down in the lab, let's see if we can actually breaking bad in the lab. And when I'm talking about breaking bad in the lab, I'm talking about the fact that we can take the BBB and the neurons, the blood-brain barrier and the neurons. Here you can see human brain microvasculature. Here you can see human neurons. And then we can induce crystal meth, methamphetamine. We can see how actually it opens the blood-brain barrier. And here you can see holes in the vasculature. And here you can see that on the right in the magenta, that suddenly you have a lot of waste that comes from the blood side and actually staining the neurons and sticking to the neurons. And this is actually what happens to drug addicts that use crystal meth. So if you need to remember something from this talk, is that don't use drugs, because it will mess up your brain. But additional things we can do with it is actually to use the fact that the system is linked, and we can actually bring labeled glucose to the blood side, similar to the fact that you will take glucose from your orange juice. It will go through your blood, but since the glucose is labeled with C13, it will start to metabolize. We will have the pyruvate and the lactate, and we will have the whole TCA cycle, and we are able to monitor by using mass spec how each one of these metabolites was formed from this glucose. And we can do it not only in the vasculature side, but keep in mind that we can actually trace how this glucose was metabolized during the different compartments. So we can put here the glucose, but to see how it was metabolized in the brain, the BBB, the prevasculature, and all these different parts, actually enables us with the metabolomic, it enables us to identify a unique metabolic coupling between the BBB and the neurons. And we were able to do some bioinformatics to actually identify the different pathways that are changing, that are significantly changing due to this interaction between the BBB and the neurons. Other things we can do with the brain on a chip, we can actually mimic different disease. For example, here in vivo, we look at newborn meningitis. We actually did the same in vitro. We were able to take newborn meningitis that was isolated in Budapest at the 70s, and to bring this to our system. Here you can see neurons, and here you can see the BBB. And what you can see here is the control of the BBB. In what you have here in red is the tight junctions of the BBB, and nothing is changing, as you can see, over time, because this is the control. So I'm actually very happy. Nothing is changing. But when we add the bacteria, which are the green dots, you can see immediately that the red staining decreases or vanish. And this is due to the bacteria that actually destroy the tight junctions. We can use actually the confocal to identify how the bacteria is penetrating through the BBB. We can do Z-stack and to see how actually the bacteria is penetrating through the BBB. And we can take our samples to scanning electron microscope and actually to see the invasion of the bacteria to the endothelium. We can do the same to neurons, to stain the neurons. And here again, over time, you don't see a significant change. But when you add the bacteria, you can see that the red staining actually decreases significantly. And the red are the neurons, the blue are the astrocytes. And we can also use the different sensors we have to monitor the electrical activity of the neurons. And you can see here that the neurons don't change significantly their electrical activity for the control. However, when we add the bacteria, you can see that the electrical activity is decreasing significantly already after 4 hours, and nothing actually stays active after 24 hours. So we can actually mimic different type of disease also using the chips. But as I said, organs on a chip is a cool tool, it's great. However, I want to be honest with you, and it's not that easy to work with. You need to have a lot of know-how. You need to have specific tools, specific pumps. Usually you can either fabricate the chips or you need to buy them from companies. And our vision in our lab is that things need to be accessible. And if we will not make sure that our technology is accessible to other labs, it will stay in my lab and nobody will benefit from this. So our mission is basically to somehow translate organs on a chip that will fit standard lab tools. And the way we do it is actually to take a standard well and to integrate this with the chip that we developed, that we 3D print. And once we insert the chip to the well, we create this organs on a chip that is that can fit to any standard lab tool. In other words, it is a very, very modular tool that can fit any standard lab tool that can be modified the way you want because we 3D print the chips. And here you can see different geometries, straight line, or other examples as well. Since we 3D print the chip, we can play with the height of the chip, with the size of the chip. You can see it fits 6 well, 12 well, 24 well plate. We can link the chips and to create multi-organ linkage. We can use commercial sensing systems such as TEER to measure permeability, such as MEA, multi-electrode array, to measure neuronal functionality. We can co-culture up to 3 different cell types, one on top of the membrane, one under the membrane, and one on top of at the bottom of the well. So here you can see we have 3 different cell types: UVEX, sushi, which is a neuroblastoma, and the glioblastoma U87. What else we can do with the chip? We can actually combine this with the super resolution microscopy. And the chip enables us to culture multiple cell types. Then we can cut the membrane in a special way. And we can use super resolution microscopy to image multiple cell types that are actually more than 2D. Why it is important? Because super resolution actually gives you the added value of being able to image things in that are beyond the diffraction limit. However, usually you are super limited by the working distance. The working distance is usually no more than 170 microns, but these chips enable to work with two sides, so you can multiply the working distance in that sense. So here you can see that we have UVAC and U87. Here we were able to image mitotic spindles, and this is another nice image that we have that we were able to use the super resolution. And you can do many things with this. Again, I'm here to show you the tools that we do and the things we do. However, we are very modular, and we can definitely modify our technology to your needs. So if you have a specific need and a specific challenge, we are the first one who will be happy to tackle this challenge and to build something that is relevant to what you're struggling with. Here is an example that I'm collaborating with Professor Uri Asheri, who is studying the alpha-synuclein aggregation, and we use this chip actually to see how alpha-synuclein, which is the biomarker for Parkinson's disease, how it is actually propagating from one type of population to another type of neuronal population. And the way we did it is by taking donor cells that actually overexpress alpha-synuclein. Here you can see with the super resolution that we can actually look at the alpha-synuclein in a very high resolution. And then we, at the bottom, under the membrane, we have acceptor cells that are healthy cells which do not overexpress alpha-synuclein. And here you can see that we can actually monitor the alpha-synuclein that is being here aggregated by the overexpression of the alpha-synuclein. But also we want to see if the alpha-synuclein actually propagates from the upper population to the lower population, to the bottom. And here you can see, again, with the super resolution microscopy, that we can look at the aggregation of alpha-synuclein. So to summarize this part, we can actually use the organs on a chip for many things. We can use it to study how drugs actually affect the brain. We actually study traumatic brain injury. We develop smart chips and smart organs on a chip, and we study how the interactions, organ-organ interaction using different organs on a chip. But at the end of the day, you can definitely use your imagination and to do it basically whatever you want. And if you have currently a specific challenge, we will be more than happy to help you to tackle the challenge. And as I said, now I hope it's clear what I meant at the beginning, that organs on a chip are a very complementary tool to the IPS world that you just saw. Because in the IPS world, we talked about, or the other people talked about the relevance to the human tissue, but you need to keep in mind that our system is very modular, and it can contain any cell type you want from any source. So in our organ on a chip system, we use cell line, we use primary cells, we use iPS cells, we use brain organoids, we use different things, and you are not limited to a specific cell type or a cell source. Our vision is that we will have a specific patient transform from IPS to specific tissues, and we will create like a mini me on a chip, which involves different IPS systems. And I cannot talk about, in such an amazing conference, which is focused on IPS, I cannot talk a little bit about the IPS work that we do. Actually, we established, Uri Asheri, Professor Uri Asheri and myself, we established here IPS differentiation core facility that we give a service for differentiation. And what we currently do in our center, we work on Parkinson's disease. And here you can see human dopaminergic neurons from Parkinson patients. We work together with Dr. Yael Aberman, and we have created gut organoids from patients with colitis. We work on familiar dysautonomia, and we create here, these are human PNS, peripheral nervous system, human iPS-derived PNS cells. And here you can see the staining of the cells. We work on kidney fibrosis together with Benny Deckel, and we create kidney organoids to look at the fibrosis. And what you see in green is basically collagen, which create fibrosis. We work with Dalit Ben Yosef about Fragile X, and to see how Fragile X mutations actually change neuronal functionality. And also, we create isogenic models of the neurovascular unit, and we use the brain microvasculature. To test different drugs and different and basically to use the low permeability of the brain microvasculature that we fabricate in the lab to test different things relevant to the brain. And this is led by Dr. Yael Bardugo from our lab. We use our models and our system for many things. We study COVID, and we have some papers about COVID. We work on neurodegenerative disease, and we also work on cancer treatment. But as I said, we are very, very versatile in what we do. So basically, the organizers told me to aim for 45 minutes talk, and to give about 10 minutes 10 minutes for questions. So before I will finish, and I will use my last 10 minutes, I would like to show you something completely different. Now, when I say completely different, I'm talking really not related to the IPS world. However, it is still connected somehow to organs on a chip. But I thought it would be a nice way to finish my talk. So all of you probably know this thing. This is the metal detector that you have at the entrance to the airport, and it costs a couple of $1 million. You know, it is super sensitive. This is basically the cutting edge technology to identify what you carry when you go to the airport. But all of you know that once you pass this metal detector and you run to the gate or to the duty free or whatever, and suddenly something can jump on you and to say that you carry drugs. This thing would be a dog. And why they will use dogs is because dogs and other animals have super sensitive sensors. And actually today we cannot compete with some of the sensing capabilities that we have at the animal kingdom. And think about it, it can be actually really, really cool if we can integrate the biological sensors with robots, and therefore to create something like biohybrid robots. Just to give you a sense of how sensitive animals are. So when we talk about insects, their ability to sense is extremely high. They are super robust, super energy efficient, very lightweight, and very, very efficient. And usually the best example I can give is think about a mosquito, right? So the female mosquito actually suck your blood or hunting us by their ability to identify the amount of CO2 that we emit compared to the amount of CO2 there is in the air. So just to remind you, the amount of CO2 we have in the air is 0.03%, which is extremely low. And yet the mosquito can identify our blood by identifying the amount of CO2 that we emit compared to the amount of CO2 we have in the air. So it's very impressive. So imagine if we can take like the nose of the mosquito and to use it for a CO2 detector, it will serve both as killing mosquitoes, which is obviously something beneficial, and also it will give us a very, very sensitive CO2 detector. So we didn't do this yet, but what we were able to do as a collaboration with Professor Amir Ayali and Yossi Yovel, we were able to take the ear from a locust to create a ear on a chip and to integrate this with a robot. And by this, we created a robot that can actually hear with a biological ear. Right? It can listen, it can identify sound using a biological ear by using the ear on a chip system. When we clap one time, the robot will identify the sound and will move. And we will clap two times, the robot will identify the sound and will move backwards. And this is what you see. I don't know if you can hear the sound at the auditorium. I clap twice, the robot is moving. Here at the tip, you can see the biological ear. And when we clap one time, the robot listen with the biological ear and move to the other side. And now I will ask the question, what a biological ear on a chip integrated with a robot is good for? The answer is nothing. It's not really good for anything. We don't really need a biological ear connected to a robot. We have excellent microphones, so there is no really added value of the chip itself connected to a robot. However, there is a huge added value by showing a proof of concept that we can actually take the sensing part of an insect and to integrate this with a robot to create biohybrid robots. Where there is actually a huge added value is when we talk about smell, because the ability of the ability of insect to smell things is much greater than any electronic device that we currently have. And it's extremely important when we think about drugs, explosives, and just the need to identify smell. So what we did, we took the ear part, the smell part, the antenna, which identifies smell of a locust. We created the smell on a chip. We were able to induce smell to these specific antennas. We induced this smell known as benzaldehyde. Although it sounds really a horrible smell, actually this is the smell of marzipan. And you can see here a really nice graph. Then we added citronelle, which is another smell of citrus. And we were able to induce many different odorants. And what is nice in this graph, and this is the only thing you need to keep in mind or to take a message from this slide, is that you see that each line has a different shape. So we were able to use AI, machine learning algorithms, to identify and discriminate odorants. We were able to do it up to 20 different odorants already. And this project was led by Neta. She's our PhD student. And we're actually extremely happy. We are super happy that we can identify and discriminate odorants. And then I came to my students and I told them, you know what? I have secret samples for you. And I want to see if you can identify, if you can discriminate between the smell. And they came back and they show me that here are my 6 samples. And I get different spectra for each one of my samples. And then I show them my different vials with different orders. And they were happy to see that they were able to get these results, but they were curious to see what these samples are. And then I showed them that actually they can use the system to identify the unique smell of whiskey. And as you can see, different whiskey, if it is from a different brand or different year, can produce actually a different smell. So with keeping that in mind, I will show you that what we were able to do is actually to put this ear on a chip, this smell on a chip system on a robot. Here you can see that Neta comes with the smell. The robot actually can smell what the specific order, and when we give a control, it doesn't move because it doesn't identify the specific smell. It's not also, the antenna has mechanoreceptors, so we just want to make sure it's not due to air. And you can see that our robot is actually moving towards a specific smell. So with this, I will finish here. This is the time that they told me to finish. So here I would like to thank the team, because actually at the end of the day, I'm not doing anything in the lab. There are the people who do all the hard work. I would like to thank the funding agencies, the Israeli Foundation, ERC, the Israel Science Foundation, Oftzin Zimin, and Teva, and others. as I told you, we collaborate with many people, and because we don't like to keep the technology in our lab. So if you are interested collaborating with us, you are actually more than welcome. We are really eager to share the technology. And as I said, we have different topics we're working on. I didn't show all of them due to the time I have, and I didn't want to put too many slides. You are more than welcome to come and visit us. We have great beaches, great food. You are more than welcome to come. And you know, you are more familiar with me probably with the term, it's always 5 o'clock somewhere. So now it's 8:45 on your side, but here it's 5 o'clock. So in that case, while you're going to grab your coffee now, for me, it's time to grab a whiskey or a beer. So, you know, I'm in a good vibe right now in that sense. Jokes aside, thank you very much again for inviting me. And if you are interested to come to Tel Aviv, we are looking for PhDs and postdocs to join our team. And thank you very much again. Let's see, I already see we have a question. Okay, so I will read the two questions I have. Thanks, excellent work, excellent talk. First of all, thank you very much. It feels like science fiction's come true. Thank you very much. I sure hope so. You mentioned 60 to 90% of the drugs pass rodent tests, fail in clinical trials. With organs on a chip, what is the passing rate do you estimate? Do you think organs on a chip will replace rodent models in the future, at least in drug development field? Well, Song, thank you for the question. It's actually a really nice question. My answer will be as follows. First of all, the statistic depends on the type of field of drug that you develop. For example, cardiac, the success rate is a little bit lower. Neurology, it's a bit lower. Cancer, it's a bit higher. So the success rate depends on the type of field the drug is being developed to. Regarding how good organs on a chip will be regarding replacing animal models. So obviously I show you the good things. There are still many challenges. And for example, there are still many systems that we still need to develop that will be more robust. For example, the endocrinic system, the immune system. So there are many systems we need to improve. However, the FDA already examined the request to use organs on a chip as a tool for use for drug development. And for now, drug companies actually started to work with organs on a chip. You can actually go and see different papers talk about the fact that as more and more drug companies publish papers that actually they use organs on a chip and they use it in parallel to rodents. And hopefully in the future, yes, it will replace animal models, but currently it is in parallel too. So this is the question that was by Song, by William Butcher. Sorry if I didn't pronounce your name correctly. Amazing work. Thank you very much. I love that you can cut out the chip membranes so you can do super resolution microscopy. A lot of microscopes have these kind of issues, working distance, exactly which limit. Another example would be holotomographic imaging, completely right. Have you been working or any way to do so? Fancier imaging, modulating with your system without having to cut the membrane. Yes, yes, we did. We don't really need to cut the membrane. Actually, we have this really cool device that can actually, as is, can go, because at the beginning, when we actually started to cut the membrane, actually it really changed the level of the membrane. So the membrane, in order to do super resolution microscopy and different type of microscopies, you need to have a membrane that is really flat. And if you start to cut the membrane or to do different things to the membrane actually will twist and will be with many small hills, and the imaging will be really bad. So actually, yes, now we have a special holders that you don't need to even to cut the membrane. So this is related to your question. So thank you. And now if there are any questions from the audience, I'll be more than happy to. Here, I don't know if you can see my backside, but here I have my whiskey already in the office. Here you can see.
Dr. Maoz,
My
I'll
bunch of whiskey
ask
over
a
there.
couple
So I'm
quick
ready. Here it's 5 o'clock.
questions so you can go to your whiskey sooner. It's really amazing work. I'm surprised you don't have an army working for you with all those different things you're working on. So you listed all those collaborators and things, and it looks like you're designing and building some of the chips from scratch in your lab. How long does that generally take? So when someone come to you with a request?
Actually, it's a really good question. So first of all, it depends on the how challenging the problem is. Most of the times we try to go to the easier solution so that it will be easy also to implement afterwards. Because if it is something that takes a lot of time and then you cannot use it, it's not beneficial for us and for the collaborator. Most of the time it takes actually really quick for example, to modify the first chip that we did to be able to do super resolution, it took us between 1 to 2 months, and then we were able to do it. So it's not that much of time. To integrate different sensors, it took us a little bit more time, but it depends on the need. But as you saw, we are really eager to collaborate, and people can come to our lab. We already had more than 20 people in the last 2 years visiting our lab from the US and from Europe. So again, more than welcome to.
So are you like turning any of these into actual products?
Yes, actually, yes. Currently, we are, first of all, yes, we have IPs on some of the chips. We have also some commercial, different companies are interested to actually put this in market. And basically, yes, we did. And yes, we are. But since we talk about academic collaboration, for this, we were more than willing to share.
Last question for me is, so you are going to be the first lab that connects different chips together to make a true microhuman soon?
So first of all, obviously we inspire to be the first in different things we do. There are the organs on a chip world. There are many different labs and many companies actually that started since the early days at the Visa Institute, and together with Anna Herland and other people, we were able to push forward to do many organ linkage experiments. But currently, it is still a great challenge. It's a huge challenge to try to do IPS of multi-organs. Currently, one lab was able to do up to 4 different organs with the IPS, and definitely this is something that we are eager to help and to push forward as well. You have a lot of energy, like 9 a.m. to run from one side to the other side. It's a lot of energy in the morning.
It's the coffee. The whiskey will have the opposite effect eventually.
Yes, eventually.
I'm Kelly Van Dussen. I was wondering about your magnet with the kind of contraction of your cardiomyocytes on the chip, right?
Yep.
So does the speed at which the magnet moves actually affect the kind of behavior of the cells or their fate later?
Yes. So thank you for the question. Actually, it's a really because every time that you build a talk, so you need to make a balance between how much you show from each project and how broad you want to be, right? So obviously, I didn't show a lot of data we have, and what you just mentioned is extremely correct and true. It is extremely relevant, the velocity of the magnet. One second. So it's extremely important, the velocity of the magnet, and it usually need to be synchronized with the electrical stimulation that we do. But also the rate, like, if we stimulate this in 1 hertz or 0.5 hertz or 2 hertz, obviously there is an effect of things we see. but also the amount of bending we give. The amount of force that you apply has a significant effect on the results, significant. And also it's also like this in vivo, but if you pass a certain threshold, you actually overstimulate the cells, you overstretch them, and actually you injure them. So you actually want to be in a certain regime that you don't stretch it too much, but you still apply a sufficient amount of pressure or shear stress on the cells. Yes.
Great, thanks.
Any other questions? All right. Thank you again, Dr. Maoz.
Well, thank you again for inviting me. And again, you're more than welcome to come and visit us here at Tel Aviv. And if you're interested to join our team, you're more than welcome to send me an email and either to collaborate or just to come and to join and to visit us. So again, thank you very much.
Thank you.